Hello and welcome to this lesson for Advanced Higher Biology, and this one is going to be looking at phosphate binding. So previously we were looking at ligand binding and how that can affect protein conformation. What we're going to look at today is phosphate specifically as a ligand and how it can be reversibly bound to different proteins. And next we'll be moving on to look at the movement of different molecules across the plasma membrane and how these molecules use proteins. So what we're looking at is learning how additional molecules can bind to and affect the protein. And what we're looking for you to be able to do is state how adding or removing phosphate groups can affect a protein, naming and explaining the enzymes that are involved in addition or removal of phosphate, and explaining how protein activity can be regulated through phosphorylation. So when we look at phosphate as a molecule, we're looking at a phosphate group in particular. And the addition or removal of phosphate can cause what we refer to as a reversible conformational change in the protein. And it's one of the most common forms of post-translational modification. Phosphate, as you can see from this diagram here, is quite a highly charged molecule, and so when it binds to a protein, it's going to alter the interactions between the R groups, and consequently that's going to alter the shape of the protein and alter what it's capable of being able to do. Now that phosphate itself is going to primarily come from ATP. That's our main source of phosphate within the cell. And you'll have seen diagrams like this previously during National 5 and during Higher, where we're looking at moving from ATP and releasing energy and forming ADP. And as a result, we can also carry out respiration and add phosphate onto ADP, and that stores our energy. Now, that we're at advanced higher level, we're not just thinking in terms of energy, we're thinking in terms of what this phosphate molecule specifically is actually able to do. So there's a particular group of enzymes called protein kinases, and they are catalyzing the transfer of a phosphate group to a protein. So the terminal phosphate of ATP, or a final phosphate, the third one in the chain, is going to be transferred to an R group of an amino acid. So if this was our protein here and we had an amino acid towards the end, it tends to be a hydroxyl group that can accept a phosphate. Others can too, but it's usually there's a group of 3 or 4 different amino acids which tend to be the phosphate acceptors, but it's usually a hydroxyl group that's going to accept that. And so this enzyme is going to add that terminal phosphate onto this R group of this amino acid, and as I said, because it's so highly charged, this phosphate group is going to have an impact on all of the interactions between the different R groups in this protein. Remember, all of those different R groups are bound together by different interactions between them, your London dispersion forces, etc. So those impacts on the interactions are going to change the shape of the protein overall. This reaction itself happens to be reversible, and that's what makes phosphate so important as a ligand and what makes the binding of phosphate so important to proteins. If we remove that phosphate, we transport our protein back to normal and we release that phosphate back into the cell to be used elsewhere. Another group of enzymes, the protein phosphatases, are going to be involved in catalyzing the dephosphorylation reaction, so removing it from the protein and releasing it back into the cell. This will return the protein to its original shape, so that conformational change, the change in its shape, will be reversed and it will go back to normal. So the activity of a lot of different cellular proteins is regulated this way. In this diagram you can see that we've had activation by this process, so adding the phosphate molecule by the protein kinase onto our protein has resulted in an active enzyme and released a signal. Now, it is also the case that it might happen the other way around, so we may have it already switched on permanently, the enzyme might be active, and actually the addition of the extra phosphate would turn it to be inactive. It's not always the case that phosphorylation is an activity signal. It can be the opposite, it can switch something off or cause something to stop in its process. The resultant reversing change, if we did want to switch that enzyme off, catalysed here by protein phosphatase. And say if we remove that phosphate molecule from our protein and release it back into the cell, we now have an inactive enzyme. So that's the importance of it being reversible, is that we can switch on and off different proteins depending on the different conditions in the cell. So the signals that are generated by hormones or neurotransmitters are often carried from the plasma membrane itself through phosphates interacting with those proteins that are embedded in the plasma membrane. That's going to have a cascade effect. It's going to cascade through different proteins within the cell, going from the plasma membrane to the nucleus as that phosphate moves on and changes the conformation of different proteins. One major example of this is when we have the regulation of protein action in terms of the muscle proteins. So we have actin and we have myosin here, which are two main muscle proteins. You may see this referred to quite a lot if you look at any old past papers. You don't need to know about their structure specifically, but it's a common example of using phosphorylation to activate proteins and regulate their action. If we look at the actin and myosin molecules here, the phosphorylation causes a conformational change in both of them, and you can see the myosin filament is changing its shape here and is adding on. Essentially, the proteins are being caused to contract when we have those phosphate molecules attached, and the release of the phosphate molecules therefore causes the muscles to relax. So that sequence of events is often referred to as a cross-bridge cycle. You may come across that if you're reading up anything. You don't need to know about that specifically, but just to be aware that that's what they're talking about, just because of this connection between the actin and the myosin, different muscle proteins there. This can be referred to as a cross-bridge here. So when we have those phosphate molecules acting on these proteins, it changes the shape and allows muscles to contract, and then removing the phosphate is going to change their shape again, allow them to relax back to the state they were in previously. So muscle phosphorylation is a good example of the regulation of protein action by our phosphates. So you should know that addition or removal of phosphate groups can affect the shape of a protein depending on the charges between the different R groups of the amino acids, that the enzymes protein kinases are responsible for the phosphorylation of molecules, so they're going to add our phosphates to proteins, and that the enzyme groups protein phosphatases are going to be responsible for the removal of phosphate and returning the protein back to its original state. The protein activity can be regulated through phosphorylation by phosphates either being added to switch on or switch off different proteins, should they be enzymes or hormones or neurotransmitters or anything of that sort. As ever, if there's any questions, let me know and we'll be sure to go over this in class later on.